Today's class is on bacteriophage. What is bacteriophage? They are viruses that infect and parasitize bacteria. They are also abbreviated as phages. Tuart and Daguerre, they independently discovered bacteriophage. Daguerre observed some invisible minute parasite of bacteria that causes lysis of the culture of dysentery causing bacilli. Hence, he suggested that the lytic agent was a virus and he named it as bacteriophage. Definition of bacteriophage is they are obligate intracellular parasites that multiply inside the bacteria and by making use of some or all of the host biosynthetic machinery. These phages occur widely in association with bacteria in the environment and are isolated from feces, sewage and other natural sources. Coming to the morphology of bacteriophage, phages that attacks E. coli are called as coliphages and are also called as T-even phages, which have been studied in details. The phages are tadpole shaped and it have a hexagonal head and a cylindrical tail. The head of phage is hexagonal in shape and it contains a tightly packed core of double-stranded DNA as a genetic material, which is covered by a protein coat or capsid. The size of the head varies in different phages that ranges from 28 to 100 nanometer. Tail is cylindrical in shape and it is consists of a central hollow tube which is surrounded by a contractile sheath and it has a terminal base plate. This terminal base plate is attached with tail fibers. These tail fibers are usually 6 in numbers. Each corner of the base plate, it contains a spike or short pin. Life cycle of bacteriophage. There are two categories of bacteriophage based on their life cycle. They are virulent or lytic cycle, temperate or lysogenic phages. In lytic cycle, the virus injects its DNA into the host cell, that is the bacteria. So once after ingestion, the host cell starts copying the viral DNA and other accessory parts of the virus. So once it is done, the phage causes lysis and death of host cell by releasing new viruses. But in lysogenic cycle, after the DNA is inserted, the phage integrated into the host cell DNA and they do not cause any harm to the host cell. Coming to the lytic cycle, the replication cycle of virulent phage is divided into 5 stages. They are adsorption, penetration, synthesis of phage components, maturation and release of progeny phage particles. Adsorption. It is the first step that takes place in the replication of the phage. What is meant by adsorption? It is the attachment of phage to the bacteria. This occurs when a phage collides with the host bacteria and it is a specific process. Here the proteins which present in the tail of the phage helps to bind a specific receptors which is seen on the surface of bacterial cell wall. For this adsorption step, certain cofactors are also needed. What is transfection? It is the infection of a bacteria by the naked phage nucleic acid. Next step is penetration. Following attachment into the bacteria, the phage injects its DNA into the cytoplasm of the bacteria. This takes place when the tail sheath contracts and the tail fibers of the virus that bend to bring the spikes, and then the basal plate comes in contact with the surface of the bacterial cell wall. Now the contractile sheath is pressed on the bacterial cell wall and the viral DNA is injected into the cytoplasm. This step requires energy from ATP. So once after penetration of DNA into the cytoplasm, the empty head and tail of phage will remain outside the bacteria. So it is called as ghost. What is meant by lysis from without? If bacteria are mixed with phage particles at high numbers, phages produces multiple holes on the bacterial cell wall. So that causes leakage of cell contents. So bacterial lysis occurs without viral multiplication. So this process is called as lysis from without. Next step is synthesis of phage particles. Once after the ingestion of viral DNA into the bacterial cell, first the early proteins are synthesized. Early proteins are nothing but enzymes that helps in repairing the holes which is produced by phages in the bacterial cell wall, and it is also necessary for building the complex molecules of the phage particles. After this, the late protein appears that includes protein subunits of phage head and tail. During this process, the synthesis of bacterial DNA, protein and RNA stops. Next step is maturation. In this step, the phage DNA, head protein and tail are synthesized separately in the bacterial cell. Then the DNA gets condensed into a compact polyhedron and packaged into the head. Finally, the tail structures are added. Maturation is assembly of phage components into a mature infective phage. Last step in Lytic cycle is release of progeny phage particles. Here, the bacteriophage produces certain enzymes that helps in break down the plasma membrane and cell wall of bacteria, causing it to lyse. So, cell lysis occurs, which releases hundreds of new phages which can find and infect other host cell. What is eclipse phase? It is the time period between the ingestion of viral nucleic acid and the first appearance of phage. During this period, viruses cannot be detected within the host cell. The duration of eclipse phase is about 15 to 30 minutes. What is latent period? It is the time period between the injection of viral nucleic acid to rupture host cell. The entire process, that is from the adsorption to the appearance of new virus, takes about 40 minutes. Rise period. During this period, the number of infectious phages released also rises. Burst size. It is the number of virions that is produced per host cell is specific. Generally, it is 200 to 300. Next, we will see about lysogenic or temperate cycle. In this cycle, it allows a phage to reproduce without killing its host. The first two steps, that is adsorption and penetration, is similar to Lytic cycle. So, once the DNA is inserted into the bacteria, the phage DNA gets integrated into the host cell DNA. The integrated phage genome in this state is called as prophage. This prophage, it behaves like a segment of the host chromosome and it replicates along with it. That process is called as lysogeny. The bacteria which carries a prophage is known as lysogenic bacteria. In this cycle, each time a host cell divides, the prophage is also copied along with the host DNA. There are certain conditions that make the prophage to convert into virulent lytic phage. This can happen spontaneously or by some physical or chemical agents such as UV rays, hydrogen peroxide, mustard gas, etc. What is meant by lysogenic conversion? Some lysogenic phage, they carry certain genes that can enhance the virulence of the bacteria. Example, some phages carries genes that encodes toxins. So, if these genes get integrated into the bacterial chromosome, they causes the harmless bacteria to release toxin that can causes disease. Example, Corynebacterium diphtheriae. This bacillus produce toxin only when it is infected by phage beta. What is meant by superinfection immunity? If a lysogenic bacteria is resistant to reinfection by the same or the related phages, that process is called as superinfection immunity. Coming to the applications of phages. Phage assay. It is done to quantify the number of phage particles present in a solution. When phages are applied on a lawn culture of a susceptible bacterial strain, after incubation, clear zones of lysis appears. These clear zones are called as plaque. Under optimal conditions, single phage particle produces single plaque. This is useful for titrating the number of viable phages in a bacterium. Next, phage typing. The specificity of phage bacterium interaction is used for the identification and typing of bacteria, and it exhibits different degrees of host specificity. They can lyse all members of bacterial genus, species and subspecies, and it is also used for intraspecies typing of bacteria. Example, Salmonella typhi and Staphylococci. What is bacteriocins? A group of highly specific antibiotic-like substances which is produced by certain strains of bacteria are called as bacteriocins. These bacteriocins are active against other strains of the same or different species. Usually they are protein in nature. Examples of bacteriocins are colicins, pyocins and diphtherisins. Colicin is produced by E. coli, pyocins by Pseudomonas pyocynaceae and diphtherisin by Corynebacterium diphtheriae. This by bacteriocins, they resemble bacteriophage by adsorption onto a specific receptor and morphological similar tail structure. This bacteriocins, it will kill the cells without lysing it. Thank you.